What if everything you thought you knew about your ancestors, the stories whispered across dinner tables, and the heritage etched into your very identity, was fundamentally based on a biological misunderstanding? Imagine opening a nondescript envelope from a genetic testing lab, expecting a confirmation of your deep Cherokee roots, only to find a map that points exclusively to the cold shores of Scandinavia and the rolling hills of the British Isles. For thousands of people every year, this isn't a hypothetical scenario. It is a jarring, soul-searching reality. We have been raised to believe that DNA is an unbroken chain, a perfect ledger of every person who came before us. We view it as a mathematical certainty, an archive where every drop of blood counts for something. But the truth is far more spectral and far more elusive. Genetics is not a ledger, it is a lottery. You can be the direct biological descendant of a historical figure and yet carry absolutely none of their DNA. This paradox, the gap between our genealogy and our genetics, has created a modern identity crisis, particularly in the United States, where the Cherokee princess narrative has become a cornerstone of family folklore. To understand why so many family legends vanish under the cold light of a laboratory sequence, we have to look past the colorful pie charts and zoom into the chaotic mechanism of inheritance itself. We often imagine our ancestry as a river, where the waters of our four grandparents mix perfectly into our parents and then into us. We assume we are exactly 25% of each grandparent. If a great-great-grandmother was a full-blooded Cherokee, the math seems simple. You should be 1/16th, or roughly 6.25% Indigenous American. But biology doesn't do math with a calculator. It does it with a deck of cards. Every time a child is conceived, a process called recombination occurs. During the formation of eggs and sperm, the chromosomes inherited from the mother and father don't just sit by side. They embrace, crossing over and swapping chunks of genetic code. It is a biological shuffling of the deck. This means that while you definitely inherit 50% of your DNA from each parent, the distribution of who those parents inherited their DNA from is entirely random. You might get 30% from your paternal grandfather and only 20% from your paternal grandmother. Over the course of 5 or 6 generations, this randomness becomes a scythe. Genetic segments from a specific ancestor can be thinned out, bypassed, or deleted entirely from your lineage. You possess a genealogical tree that includes thousands of ancestors, but you possess a genetic tree that includes only a fraction of them. This brings us to a startling realization. There are people in your family tree who are your ghost ancestors. They are your direct forebears. You wouldn't exist without them, but they have left no physical trace in your genome. When we talk about the search for Cherokee DNA, we are often searching for ghosts. The tension between the stories we tell and the molecules we carry has created a multi-million-dollar industry, but it has also exposed a massive rift in how we perceive race, culture, and belonging. We see the DNA test as a truth machine, but what if the machine is answering a different question than the one we are asking? The narrative of the lost Cherokee ancestor is one of the most persistent tropes in American genealogy. It is so common that professional genealogists have a shorthand for it. For many, the discovery of 0% indigenous markers feels like a betrayal of a grandmother's memory or a theft of identity. But to solve the mystery of why these results are so consistently European, we have to move away from the microscopic level for a moment and look at the macro level of history, migration, and the brutal reality of the American frontier. The DNA isn't just a record of who slept with whom. It is a record of who survived, who fled, and who chose to disappear. As we peel back the layers of these genetic results, we find that the surprise isn't usually a mistake by the lab. The surprise is a reflection of a complex social camouflage that took place centuries ago. In the 18th and 19th centuries, the racial landscape of the American South was a dangerous hierarchy. For many families of mixed heritage, specifically those with African or indigenous roots, identifying as Cherokee was often a safer, more socially acceptable explanation for darker skin or exotic features than the alternative. In a society governed by the one-drop rule, where any African ancestry could mean enslavement or the loss of basic rights, a Cherokee ancestor became a vital piece of folklore, a shield. But this discovery revealed a hidden cost. The stories we used to survive the past have become the errors of our present-day genetic tests. To understand why a DNA test might show Scandinavian or Irish when your family history screams Cherokee, we must confront the mechanism of the genetic bottleneck. The Cherokee, like many indigenous nations, were not static, isolated populations. Long before the advent of modern sequencing, the interior of the international Southeast was a crucible of interaction. Traders from Scotland, Ireland, and Germany pushed into the Appalachian backcountry as early as the late 1600s. These men, often fleeing their own upheavals in Europe, integrated into Cherokee society, marrying into the tribe and raising children that were culturally, politically, and legally Cherokee. Because the Cherokee are a matrilineal society, the children of a Cherokee woman and a Scottish trader were fully part of the Wolf clan or the Deer clan. They weren't half anything in the eyes of their people. They were Cherokee.
However, from a purely molecular perspective, these unions introduced vast swaths of Western European DNA into the Cherokee gene pool centuries before the Trail of Tears. When a modern descendant takes a DNA test, the algorithm scans for informative markers, specific sequences that appear at high frequencies in certain populations. If your ancestor was a Cherokee citizen who happened to carry a heavy load of Scottish genetic markers from a trader grandfather, those markers don't transform into indigenous DNA just because that ancestor lived as a Cherokee. The DNA remains stubbornly European. This leads us to the microscopic heart of the matter, the SNP, or single nucleotide polymorphism. These are the tiny variations in our genetic code, a single letter in the billions of base pairs that scientists use to track ancestry. To identify Cherokee DNA, scientists look for SNPs that are unique to or highly concentrated in indigenous American populations. But here's the catch. There is no such thing as a Cherokee gene. There are only patterns of markers that are common among people of the Americas. Because the Cherokee history is one of intense contact and integration, the signal of their original indigenous markers has, in many lineages, been dampened by the noise of European and African genetic input. But there is a more profound biological hurdle at play, the reference panel. When you send your saliva to a lab, they aren't comparing your DNA to a legendary Cherokee chief from the 1700s. They're comparing it to a database of modern people who have documented long-standing ancestry in specific regions. If the reference panel for indigenous Americas is primarily composed of individuals from Central or South America or from tribes in the western United States who had less historical admixture with Europeans, the software might fail to recognize the specific genetic signatures of the southeastern tribes. This creates a frustrating, invisible ancestry effect. You might carry segments of DNA that truly did come from a Cherokee ancestor, but if those segments look too much like the DNA of a Northern European, or if they aren't represented in the lab's reference database, the computer will categorize them as European. It's a digital erasure of a lived history. We're essentially asking a 21st century algorithm to decode a 300-year-old social reality, and the math often fails to capture the nuance of the human experience. This discrepancy is where the emotional weight of the surprising results hits the hardest. For many, the family story of Cherokee heritage isn't just a fun fact, it's a foundational pillar of their identity. To see 0% Native American on a screen feels like a clinical invalidation of their ancestors' lives. Yet, if we look closer at the history of the 19th century,
we find that the Cherokee label was often used as a catch-all for anyone who didn't fit the binary of black or white. In the tri-racial isolate communities of the South, such as the Melungeons or the Brass Ankles, Cherokee was a safe harbor. It was a way to explain away olive skin or dark hair in a world that was increasingly obsessed with racial purity. The irony is that while the DNA might point toward the British Isles, the reason your ancestors claimed Cherokee heritage might be the most American story of all. It was a story of survival, of blending, and of creating a new identity in a landscape that was often hostile to the old ones. We are looking for biological certainty in a history that was defined by fluidity. But this discovery revealed a hidden cost. As we become more reliant on these tests to tell us who we are, we risk letting an algorithm override our actual family history. If the DNA says one thing and the family Bible says another, which one do we trust? To answer that, we have to look at the one part of your DNA that almost never lies, but rarely tells the whole story: the sex chromosomes. While the autosomal DNA tests, the ones that give you those colorful pie charts, are a shuffle of your entire heritage, there are two specific paths of inheritance that bypass the chaos of recombination. These are the Y chromosome, passed from father to son, and mitochondrial DNA, passed from mother to child. These are the genetic fossils of your lineage. Because they don't mix with other DNA, they remain virtually unchanged for thousands of years, save for an occasional mutation. In the search for Cherokee ancestry, these markers act as a high-resolution lens. If a woman carries a specific mitochondrial haplogroup, such as A2, B2, or C1, it is definitive proof of an unbroken maternal line leading back to the original inhabitants of the Americas. Even if her autosomal test shows 99% European DNA due to generations of marriage with settlers, that tiny mitochondrial spark remains. It is the signature of a founding mother. However, for many who claim a Cherokee great-grandmother, the results of these tests are the most shocking of all. Frequently, the maternal line comes back as H or U, haplogroups that originated in the caves of Ice Age Europe. This reveals the Mendelian trap. We assume that because we have a story of a Cherokee ancestor, that ancestor must occupy a specific, traceable slot in our genetic architecture. But if your Cherokee ancestor was your father's mother's father, their mitochondrial DNA and their U chromosome both vanished the moment they had a daughter or a son. They are genetically invisible in those specific lineages. This is why a 0% result is so often misinterpreted. It doesn't necessarily mean the ancestor didn't exist. It means the biological lottery didn't pick the specific markers that the test is designed to find.
We are currently living in an era of genetic essentialism, where we believe our DNA is the ultimate truth of our being. We've traded the family hearth for the laboratory bench. But as we zoom in closer, from the macro history of nations down to the micro level of individual base pairs, we see that DNA is surprisingly poor at defining culture. A person could be genetically 100% European, but raised within the Cherokee Nation, speaking the language and practicing the traditions. Conversely, a person could be genetically 50% Indigenous, but have no connection to the community whatsoever. The surprising results of these DNA tests have forced a collision between two different definitions of Native American. One is a political and cultural definition based on citizenship and kinship. The other is a biological definition based on shared ancestry. The Cherokee Nation itself does not use DNA tests for tribal citizenship. They understand something that the algorithms don't. Being Cherokee is about who you belong to, not just what is in your blood. But the mystery deepens when we look at the results of people who do have confirmed Indigenous ancestry, but still show high percentages of European DNA. This isn't a mistake, it's a record of the colonial bottleneck. During the 18th century, the Cherokee were one of the most acculturated tribes in the eyes of the U.S. government. They built Western-style homes, developed a written syllabary, and, crucially, intermarried extensively with European settlers.
By the time of the Trail of Tears in 1838, the Cherokee population was already a genetic mosaic. If you were to test a Cherokee citizen from the year 1830, you might find they were already 50% Scottish or Irish. When that person's descendants test today, 2 centuries later, that European percentage has been amplified, while the indigenous markers have been diluted. The surprising European results aren't an entry error. They are a testament to the Cherokees' incredible ability to adapt and integrate outsiders into their national fabric. The DNA is telling a story of survival through synthesis. But this discovery revealed a hidden cost. By focusing so intently on finding Cherokee markers, we often ignore the other surprises waiting in our code. For many Southerners, the search for a Cherokee princess has led to a much more silenced reality, the presence of sub-Saharan African DNA that was obscured by the same social camouflage used to hide from the one-drop laws of the Jim Crow era. This realization brings us to the most uncomfortable chapter of the American genetic story. For decades, families throughout the South and Appalachia used the Cherokee narrative as a genealogical placeholder. It was a way to explain away high cheekbones or dusky skin without acknowledging the presence of African ancestors in a society that was violently segregated. When modern DNA tests revealed 2% or 3% West African ancestry alongside 95% European markers and a total absence of indigenous DNA, the information gap is finally bridged. The Cherokee princess was often a social fiction used to protect the family from the brutal legal consequences of being classified as mulatto or black. To understand the micro-mechanics of this, we have to look at how these small percentages of DNA persist over time. Unlike the ghost ancestors who disappear entirely, some genetic segments are sticky. Due to a phenomenon called linkage disequilibrium, certain blocks of DNA tend to stay together as they are passed down through the generations. This means that even 200 years after a specific ancestor lived, a modern person can still carry a tiny, unmistakable signature of that person's origin. When the lab identifies these segments, they aren't guessing. They're looking at the literal physical evidence of a person's history, evidence that may have been intentionally erased from the family's oral tradition. This is the true power of the Helix horizon we are now navigating. We are moving from an era of myth-making into an era of biological transparency. But transparency isn't always easy to digest. When the data contradicts the story, it creates a psychological identity dysphoria. People feel as though their ancestors lied to them, or that they have been faking an identity they truly believed in. Yet, if we look at this through the lens of a world-class scientist, we see that the story and the DNA are simply 2 different types of data. One tracks the movement of molecules, the other tracks the movement of human hearts and the strategies of survival. Consider the Mendelian simplicity we were taught in school. We were told that we are a clean mix of our parents. But as we have seen, the reality is a Waddington landscape of staggering complexity. Your European results might be the result of a marble rolling down a specific path of recombination that just happened to miss every single indigenous marker your ancestor carried. Or perhaps more likely, your European results are a reflection of the fact that the people we call Cherokee or indigenous have always been part of a global, interconnected human story.
The DNA test is not a judge, it is a mirror. But it is a mirror that reflects a much wider world than the one we see in our family albums. It shows us that we are not silos of purity, but tapestries of migration. The surprising ancestry results that go viral on the internet are only surprising because we have spent so long believing in the illusion of pure lineages. In reality, every human genome is a chaotic, beautiful record of survival, adaptation, and the constant blurring of boundaries. The question then remains, if your DNA doesn't match your story, who are you? The answer isn't found in a laboratory. It's found in the realization that while our genes provide the blueprint for our bodies, our history provides the landscape for our lives. We are the stewards of both. By reconciling the two, we don't lose our identity, we gain a much more profound, albeit complicated, understanding of what it means to be human in a world that is no longer hidden by the shadows of the past. But this discovery revealed a hidden cost. As we unlock the secrets of our deep ancestry, we are beginning to realize that the most influential ghosts in our DNA might not be our human ancestors at all, but a different species entirely. If you think a 2% shift in your ancestry is life-changing, wait until you see what happens when we look at the 2% of your DNA that isn't even Homo sapiens. Would you like me to show you the hidden Neanderthal legacy inside your own cells?